The heat shock response is an essential cellular stress response pathway that maintains cytoplasmic proteostasis and can be characterized at the molecular, cellular, and organismal levels in C.elegans.
We present a series of standardized protocols and best practices that generate robust induction of the heat shock response in C.elegans in order to help enhance reproducibility in the heat shock response field.
We show that thermotolerance, a commonly used organismal assay, is not dependent on the heat shock response. Instead, we recommend using thermorecovery, an organismal assay that is heat shock response dependent.
The heat shock response becomes attenuated with the onset of egg laying. Therefore, developmental timing is a critical variable that must be accounted for in heat shock response experiments.
Begin by creating a synchronized worm population. Use a platinum wire pick to transfer approximately 10 gravid adult worms to a fresh plate. Make sure to remove any eggs or larvae that may have been transferred with the adults. Allow the worms to lay eggs for 1 hour, then remove the adults from the plate. Maintain the synchronized worms at 20 degrees Celsius until the desired developmental stage, taking into account that developmental timing varies with each strain and the temperature at which the worms are raised. When the worms are ready for heat shock, wrap the plates with paraffin film and use a test tube rack with a lead weight to submerge them in a circulating water bath at 33 degrees Celsius for 1 hour. Recover the plates from the water bath and dry them with a paper towel. Remove the paraffin film and allow the worms to recover at 20 degrees Celsius for 6 to 24 hours, which will be a sufficient time for GFP synthesis. To prepare slides for imaging, position a microscope slide between two other slides.